Thanks very much, and really grateful for this opportunity to present our work and to come back to Oxford. The last time I was here was in my 20s, visiting the first great love of my life when she was doing her doctoral work here. Exactly. So back to whole phages. At Walter Reed Army Institute of Research, which I will refer to as RARE, we have been taking the approach to try to develop fixed cocktails of whole bacterial phages that we really know that we have to work towards making much more durable cocktails in terms of how we design the cocktails. You've heard probably myriads of experts tell you that the personalized approach is the only way to go. But in the United States, the regulatory path for doing the personalized approach is difficult, as exhibited by adaptive phage therapeutics. And in addition to that, we're Army. We have the mission to try to push medical solutions out into the battlefield. So we're thinking about not only what we can apply empirically in the battlefield, but also even preventatives that can be used in combination with antibiotics, because we're thinking about what the future battle might be, and it might look more like the Pacific theater during World War II than Iraq and Afghanistan. So nothing that I say is representative of the Department of Defense or the US government. These are my own views. So we initially started with biodefense efforts. Yersinia pestis was our first target. But when we switched, when we were told we could no longer work on those pathogens, and we started working on the escapee pathogens, our first goal was to try to develop cocktails with 5 to 10 components that would address at least 70%, 75% of global diversity of the pathogen. And we wanted to leverage all the unique aspects of phage, and thinking of it very much as adjunctive therapy to antibiotic, standard of care antibiotics. And this is the pipeline that we developed over the years. So basically 5 different stages, from phage discovery and enrichment, characterizing the candidate phages, and down selecting. And this is a process of, it's like a gated tiered process where we have, parameters like host range, antibiofilm activity, the rate at which the host can become resistant to a phage. So selecting, down selecting, and then designing cocktails based on the characteristics of the individual phages, and then testing the cocktails. So actually analyzing the cocktails both in vitro and in animal models. And then beyond the animal testing phase, which is last phase of what we do at RARE, looking very much towards manufacturing and going into clinical trials. Since 2020, I was lucky enough to be able to spearhead the effort to actually involve our overseas labs. So we have a lab in Bangkok, Thailand. We have a lab in Nairobi, Kenya, and a lab in Tbilisi, Georgia, that I helped set up. And all 3 labs have been involved in phage harvesting over the last few years. This was difficult to do because we were setting it up during COVID. So all the initial training had to be done through remote means, but we did it. We have great partners. And so we've been receiving phages from them. Initially, we're going to have them do local testing to down select, but really what ended up making a lot more sense was trying to get pure phages into sequencing as rapidly as possible, and then go from there. So this is a snapshot of our our library of phages that we have collected for Pseudomonas. So we're actually up over 150 unique sequenced phages that span 17 genera. The ones you see in red boxes, we don't actually use for therapy because they're known transducers. So we tend to avoid them, but they're important for us to have because we want to avoid transduction. So they're important controls for us to have for the work that we do. The yardstick that we use to measure the host range of our phages is our diversity panels that have been developed by the Multidrug-Resistant Organism Repository and Surveillance Network, MRSN, which is a part of our institute. They do collection of drug-resistant isolates from across the entire military health system, including our deployed troops in different parts of the world, and including isolates that we receive from allies, such as the Ukrainian military. And they carefully construct these. Thousands of strains have gone into this collection. They do whole genome sequencing, and they have a core genome MLST approach that they've developed. And this slide is actually showing a new iteration. It's not new anymore, but how they made decisions about adding diversity to their original 100 strain panel to make the new one, which is available in BEI resources. So it's publicly available. So one of the things that we decided to look at from early on is the stability of phages in combination with each other. So looking at pairwise mixes of what we thought were good candidate phages for cocktails. And these are examples of those types of data. Occasionally we will see a bad actor that we want to avoid. This is stability over 6 months, or 12 months actually, sorry. And then looking at a whole cocktail combination. So this is a 7-phage cocktail where we've already done the pairwise mixes to make sure that we didn't have any problems with combinations of phages. And then resistance, because phages have co-evolved for billions of years with their hosts, we expect resistance to arise naturally. So looking at, again, at pairwise mixes and the rate at which the host can become resistant, and both individuals and mixes, and then trying to select phages and phage combinations that tend to be more resilient to resistance. The other approach, because we had done the work to establish the global collection process, was to actually take a panel of what we thought were pan-phage resistant strains from the 100 strain panel, and also from compassionate use isolates that we were receiving. So it was a 50-some strain collection that I distributed to the overseas labs, and they did collections to see if they could find phages against those isolates, and that actually helped us close gaps. So this was the final one from that project that allowed us to say that we have covered the 100 strain panel, diversity panel, MRSN diversity panel with our phage library. And that was toward the end of last year. So what I didn't say earlier is it's very much an iterative process for us of designing a phage cocktail, testing it, and then improving it, going through the whole process again. And so these are our first 3 iterations of Pseudomonas cocktails. Our PAM1 cocktail was really designed, slanted towards anti-biofilm activity. And then we found when we really started to test our phages that we didn't need to do that with Pseudomonas phages because they generally tend to have anti-biofilm activity. So you can see a shift here in the host ranges of the individual phages to much broader host range phages against the 100 strain panel. And then we have complementarity problems in the beginning that we resolved over time with the pairwise combinations. And then you can also see that we not only met our initial goal, but exceeded it by the time we got to the PAM3 cocktail, which is a 6-phage cocktail. We have some diversity. We wanted more phylogenetic diversity of the phages, some receptor diversity here, but definitely room for improvement. But this ended up being our lead cocktail that we did a lot. It's the best characterized cocktail we have. So our entry model is a wound infection model in the mouse. It's an incisional wound that's infected and then treated with phage or another antimicrobial. And we can measure wound healing over time. And also with Pseudomonas, even with a lab strain, PA01, it's very easy to tip over into septicemia and have death. So we also looked at protection against death. And this is the PAM3 cocktail. So we see with the phage group, the wounds don't get as large, and then we get more rapid closure. And with phage, we had 100% survival of the mice versus greater than 50% death in the control group. And this is pretty typical of what we see with our cocktails. We are, like I said, we're constantly trying to improve our cocktails. So this is a new iteration cocktail where we actually used in vitro directed evolution, the Appelman's method adapted. And this was the first time that we actually incorporated our phages from overseas. So this is a phage from Thailand and 2 phages from Kenya that are the parents of this component in the cocktail. And that single component replaced 2 phages in the PAM3 cocktail without a loss of lytic spectrum. So definitely a step in the right direction for us. And it also increased the phylogenetic diversity of the phages in the mix. And this is what it looked like in the wound infection model. Here we're also looking at ceftazidime as an antibiotic control and the combination of phage plus ceftazidime with either the PAM3T cocktail by itself or in combination with ceftazidime. We had 100% survival of the mice, which was ceftazidime by itself was not as good. And then more rapid wound closure. The best was with the combination therapy. A recent effort has been to work in the mouse pulmonary model, which is more difficult, but also faster and cheaper, less mice. So we looked at intranasal delivery of the PAM3 cocktail in this model using a different challenge strain. This is Paclumi instead of PA01. Comparing different routes of infection, I'm just focusing on intranasal and IV because these are the most relevant out of the ones that we looked at, particularly because IV tends to be used in the United States for everything, which we're trying to move away from. What we saw is with intranasal delivery, we have significant protection, and with IV, it's not as impressive. And it appears that part of that is the transit of the phages to the lungs takes a longer time, and that has a big impact on survival. So when we look in the for what's going on with bacterial burden in this model. This is intranasal. After 6 hours, we see a significant reduction in bacterial burden. And this particular experiment went out to 14 hours. Whereas with IV, we see a trend towards reduction, but we don't see that significant reduction in bacterial burden. Phage propagation in the infected mice, as you would expect, is higher than what you see when you're just delivering phage alone without infection. So, but we did see retention of phage in the mouse lung, even without the bacteria being there over the 14 hours. And then with and this was a significant level of propagation in the infected mice with IV delivery. We didn't see that with with intranasal delivery. We didn't see that with IV. And then this is a PK/PD study looking at 2 phages from the PAM3 cocktail. So trying to simplify things for this study, and also taking it out to 48 hours post-treatment. And so we see significant propagation, again, in the infected mice with this 2-phage subcocktail. And then in terms of CFU reduction, significant reduction in CFU when you treat with the 2-phage combination. And this persists out. The control lowering of bacterial burden persists out to 48 hours. And this is the newest work that we've done. So this is the PAM5, rare PAM5 is the newest iteration cocktail. Again, we're trying multiple methods to try to address resistance that arises. With this particular cocktail, what we did was we had done a number of experiments in animals and saw resistant strains that would arise in the infections after cocktail treatment. We used those to then isolate new phages against those resistant strains. And two of these components were phages that were isolated that way against phage-resistant strains. So trying to address the resistance that tends to arise in the model. And those 2 phages were incorporated without a reduction in host range of the overall cocktail. Actually, it's greater. We also have more receptor diversity in the new cocktail, although that's not completely fleshed out yet. More phylogenetic diversity in the cocktail, and also receptor engagement. This cocktail was one that we put together really rapidly for a clinical trial. And we unfortunately had only a 47 strain panel of Pseudomonas isolates from CF patients to use. So 4-phage cocktail went into the phage trial, which is a phase 1/2b clinical trial sponsored by NIH and led by the Antibacterial Resistance Leadership Group. And enrollment's been completed, the sites have been closed, and now now they're doing the data analysis. So this is a trial that's moved on toward completion. If we could design this cocktail today, we'd do it very differently. This was something that we put together rapidly in, I think, 2019. Just quickly looking at Klebsiella to wrap up. Klebsiella is more difficult. The phages are narrow host range in general. Our current collection has over 200 unique phages now that span at least 25 viral genera. And initial cocktail formulation was difficult. So with a 5-phage cocktail, we could only cover about 50% of the MRSN 100 strain panel, which is very diverse, 94 sequence types in a 100 strain panel. But we have composed a new cocktail that also incorporates one of our phages from Kenya, but also another trained phage, another phage that's gone through, that has multiple parents, and they were all Kenyan phages. And this cocktail is still relatively new. We're in the beginning of testing it, but over 80% coverage is a huge jump for us in coverage of the diversity panel. So I hope that I've given you a good sense of our efforts on trying to build durability into fixed phage cocktails. As I say here, it's not only the cocktail, it's also the library of candidate phages behind it that we're using, because the regulatory path in the United States, they seem amenable to switching out phages if a component becomes ineffective. To improve the cocktail and have it be more viable. Thank you very much.
Thank you very much. Amazing work, amazing work. I was just curious to know, what's the speed of resistance development? What have you been, I mean, how do you estimate?
So, yeah, I mean, it depends on how you measure it. So the one slide that I showed you were frequencies of resistance arising in vitro. One of the things that we see when we look at antibiotic synergies is, one of the problems is outgrowth and how to actually measure synergy. So it really depends on the way that you're looking at resistance.
What do you mean by the parents? Are you putting 3 phages or 2 phages together in one?
Yeah, so that approach with the modified Appelmann's, we use a number of phages that they're phylogenetically similar to each other, but they have different lytic spectra. And what we get back are chimera. And we just published on the Klebsiella trained phages. Yeah.
Great work, Mikeljon. I was wondering about the combinations of the cocktails. Do you put the phages 1-to-1 in all of them, or do you take into account some of them having like a more rapid generation time?
Yeah, we haven't gotten to that level of fine tuning. Yeah, and that's another piece of cocktail composition, that formulation that we haven't gotten into yet. Everything's been 1-to-1 so far. Yeah.
Thank you. Wonderful talk. Double question, both relating to military. Does the military directly have more freedom to use this on its military personnel? That's one part. And the second part is in terms of dual use. Will you also be able to give this to civilians in a non defense setting?
I mean, obviously, so the way that things work in my world is I always have to think about commercialization, even though I'm a government worker, because market pull is essential, because we can't manufacture it ourselves, right? So the idea is that it would be something that would be applicable to the civilian, and that would actually be the main audience for the cocktails, not just the military. But hopefully we would have, we'd hand it off to a manufacturer, they'd make the cocktail, and then we'd have it available for our troops. But yeah, in terms of we really don't have lower barriers in terms of human use. We have to deal with everything that we would in the civilian sector in the military.
I think actually on that note, unfortunately, we probably should end, but it's a interesting time to have we're sure we could all happily chat more at break. We could easily have had a